trying to get my bearings here. So let's talk about the schedule really quick. So let's see, bioinformatics exercise 1 was due Friday, so please post that if you haven't yet, but that was due on Friday. Number 2 is due this coming Friday, right?
Mm-hmm.
And then number 3 is due the 21st. I'm actually going to catch up. I've been going slow the whole semester, but I think I'm actually going to catch up, and we're going to be this week, because we are probably going to start mitochondria this week. Anyways, what I'd like to do is, just looking at the schedule, definitely give you just some in-class, a whole period maybe of in-class time to work on bioinformatics maybe on Friday, this Friday, okay? And then next, so that would be, which one is due? Okay.
2.
The one that's due is 2, so you can work on 2. And then 3, so that'll be this Friday for 2. And then next Friday, you can probably have the time to work. Well, we're gonna have a protein localization in class exercise, so we'll have some in-class time next week. So we'll get ahead, we'll get the mitochondria done so that you can just be reviewing it for the exam and getting more comfortable with it. And then we will do protein localization in class next week. And then you can have a little bit of time for 3 also, okay? For this week, I'm going to open part 1. So I'm going to do the 2 parts of the exam, and I'm going to open part 1 on Wednesday. And I'm going to open part 2, and then like you'll have Wednesday the whole time, Wednesday to Sunday to complete it. And then the part 2 will open up. I mean, honestly, they can both open up if you want to do that. Because maybe you want to do 1 and 2 and then jump back into 1 after you've done 2. Okay, so I'll try. That's my goal, is to have both exams opening up on Wednesday after class, so that you can do both of them. Do you guys have questions about? We talked about doing this part 2. I see a couple of faces that are like, I have no idea what you're talking about. So I floated this idea last week, if you were in here, to have 2 exams. One is going to be, and I've decided it's going to be chapters 6, 8, and 9 will be in the part 1. And then chapters 10, where we got really more, well, chapter 10 is protein synthesis, but 10 and on are going to be in part 2, is what I was thinking. Still kind of thinking on that though. Okay? But part 1 you're going to get 2 attempts at, so you open it, do it, you get 2 attempts at it, so you can talk. Honest, I'm okay if you're not talking during the exam, but if you get out and talk to somebody after the exam, I'm fine with that. And then you can get back in and get your second attempt in there. Make sure you get your points. And then part 2, there's only 1 attempt, so you're going to jump in and take your exam. Okay? That's the idea. That's how that's going to look. Questions about that? If you have, when you open up the information, it'll explain it all. But let me know if you do have questions. I don't mind answering, so please let me know. Okay? All right, so that's what I'm thinking. That's what I'm thinking to get us through to those last few weeks, because it's like coming up so fast. And I will update that. So we will be meeting every Friday until the week of the 24th. That Friday we will not be together on that Friday. Okay? We won't be coming in here because it's an online exam. All right. So many things open. So let's finish up with chapter 11 and 12. Oh, there was one more thing. I just saw it in here. So the key is for bioinformatics exercise is it's 1 through 3. So at the end, I updated that assignment. There's a little thing at the bottom, a little sentence at the bottom that tells you that when I'm going to post that. It's going to be before exam week, so it's the Sunday, it's the 23rd is when that key will post. Once those keys are posted, I can't take late assignments after that. All right? So you want to make sure those bioinformatics exercises get turned in on time. All right. So we have been talking a little bit about transporting proteins, and we, started off really talking about how proteins, there's different types. There are the secreted ones, the membrane-bound ones, the stuff that lives in the cytosol, and then we talked about, how they get into their unique area and they're folded properly. So co-translational, post-translational, transport into the endoplasmic reticulum, and then we went into, eventually we got to talking about their destination, moving from the endoplasmic reticulum to the Golgi, how they get sorted, what are the molecular details of that, the sugar addition, so we add some sugars in the endoplasmic reticulum, like a core of sugars, and then we make them more specific in the Golgi, and then they get packaged and sent to the right place, right? So there's a lot of pathways, lots of destinations for proteins. This is kind of where we left off. We were looking at how these different things are pinching off from the once they're really, really decorated with the proper label for where they're going, their sugar, their unique sugar combination. They are targeted to eventually the lysosome. They might be targeted to another compartment called the recycling endosome. And then they might go straight to the membrane, to the plasma membrane at the surface of the cell, and they might be embedded in that membrane, so stay there. This was also holding some secreted protein. Secreted means it's released into the environment, into the extracellular environment, and it's water soluble. Okay. I want to bring attention back to these two things because we're going to see them here in a moment again. Notice that, so this is the lysosome, and we all remember what the lysosome does, right? The lysosome is where we will send things to be degraded. So typically a lot of the food vacuoles end up fusing with the lysosome, but a lot of the lysosomal enzymes and stuff like that, they're going through this secretory pathway. They're going through the endoplasmic reticulum to the Golgi, to the lysosome, because they have dangerous things in them, right? And we just don't want them floating through the cytosol and destroying stuff, right? Destroying our cytoskeleton or something like that. So they do this. So the lysosome, a thing to remember, if you remember earlier, we talked about, bringing food in. We've talked about this in other classes too, but you get the endocytic food that gets pinched off from the surface and brought inside of the cell. And then it has to fuse with the lysosome. But the lysosome goes through all kinds of fusion. First one is, this vesicle, these things that are coming from the Golgi, they're going to go into what's called the late endosome, and then there is a lysosome. Endosome is in our picture. In our picture, it is going from, we're seeing our enzymes coming from the Golgi to the late endosome, but a missing piece of this picture is the cargo. The food cargo from the surface. So it's pinching off from the surface, and it goes into an endosome, an early endosome, and eventually it gets turned into the late endosome. And it's in the late endosome where we have the enzymes and the food come together, okay, and the acidic compartments and stuff. And it basically kind of matures into the lysosome, or it may fuse with a pre-existing lysosome to get everything that it needs. So you get all the ingredients compartmentalized in the same place. Okay? So that's a little bit complicated to think about, but you have to think about, so right now we're talking about how do the lysosomal enzymes get there from the endoplasmic reticulum to the Golgi to the lysosome. But you have to remember that other part, where is the cargo coming from? It's coming from the surface. Okay, this thing is a really cool thing too. So we haven't talked enough about endocytosis maybe, but we're seeing everything going out, but there is the inward motion of stuff. So I just mentioned the endosome. So you get endocytosis, endosome. These specific ones can also fuse with this compartment, which is called the recycling endosome. This is a really cool thing. It's not a Golgi, but it's kind of like a Golgi because it has these different sides to it. And sometimes things can come from the surface into the recycling endosome, and depending on how they are tagged, whatever their protein, their amino acid signature is, they might be sent back to the surface, which is why the word recycling is there. So they can come in from the surface into the recycling endosome and come back out to the surface. The EGF receptor does this. The EGF receptor will go inside from the surface to the recycling endosome and back to grab another ligand and keep signaling cell division. But the recycling endosome may have some specific proteins that belong in it, or it might have the right sorting capability to get some cargo to the surface. So it's sort of an intermediate from the Golgi to the surface as well. Okay, so late endosome and recycling endosome, those are two things we've seen a few times but haven't defined. So we're just defining them now. All right, how do things get targeted to where they're going? It has a lot to do with, that we've talked about what's on the protein. We add these sugars and we get a very specific sugar signature on the protein. They might find receptors, they might find some other proteins, remember calreticulin and those calcium-dependent proteins that kind of help move things in the endoplasmic reticulum and in the Golgi. But another thing is just getting it on the trans-Golgi network, getting everything that's going to a specific place compartmentalized together in the same vesicle. So here we have the apical membrane at the top of a polarized cell. And notice that the apical proteins are all coming together in this specific vesicle in the trans-Golgi network. They're going to this specific side of the recycling endosome, and the recycling endosome finally traffics it to the top of the cell, or the apical part of the cell, right? So apical means the top of a polarized epithelial cell. Basolateral is these sides. So here we are in the Golgi, we're putting those basolateral proteins in a vesicle. It goes to the recycling endosome, and they are staying on the same side of that, and they're moving to the basolateral membranes of the cell. Okay? It's interesting, even the membranes are different, right? The side membranes look a certain way, the top membranes look a certain way. They have different jobs. And that's how we define them. All right, so we've been looking at how the membranes are, how things are sorted within the membrane and move them in these vesicles. This is what? Vesicular transport. That's one of those terms we got to be comfortable with. And so how do we've talked about how, there are these transmembrane proteins, and the amino acid defines where things are going, and the sugars define where things are going. But now we are going to look at a picture of how these membrane vesicles are formed. So, you've got lipid bilayer membrane, right? And they're like this oily fluid substance. How do you pinch it off into those little vesicles? So there's protein that's helpful with that. So we have, these are some main names of ones that you will see. So there's clathrin, which is really the big, it's at play on here you're seeing the, I think that, so the membrane of the Golgi and the membrane of our cell surface, our plasma membrane. So the clathrin's gonna pinch those either side. Clathrin's gonna be involved in trying to turn those membranes into bend them so that they hold on, they grab cargo. Okay. COP1 and COP2, these are some other ones, other proteins that help to shape the vesicles. COP1 and COP2, they are mostly, do I have this in the notes? I don't. COP1 and COP2 are really in between the Golgi and the endoplasmic reticulum. They're kind of in that area. But really, it all depends on the cargo and the other proteins that the membrane-bound proteins within the vesicle bind. So there's a lot of specificity that can happen, but they can call a clathrin to them. They can call a COP1 to them. Okay? So They are cytosolic proteins typically, and like I just mentioned, the vesicle is going to have some sort of transmembrane protein that calls that cytosolic protein to it, okay, or the membrane that's going to turn into a vesicle. As long as the clathrin or COP1 or COP2 proteins are bound to the vesicle can't fuse with anything else, because it's like a protein cage all the way around this thing. Okay, so in the picture we're seeing, we have on the surface of this membrane, we've got these cool little bluish proteins that are on the membrane, and then we have some of this, they're kind of recruiting some of this water soluble stuff that floats into the vesicle. And then eventually you see, like, this is a nice and smooth one, right? We have coat assembly, and now you see that it looks like a honeycomb around it, but it's actually these little filamentous proteins that build this cage. And they actually have a shape that makes the lipid bend into them. So they actually bend the membrane.
Those are the COPs?
And then, sorry, what?
Those are the COP proteins?
COP or clathrin, yeah. They kind of look the same. Yeah, slightly different amino acid sequences. But yeah, clathrin, COP, either one of those vesicle shaping ones. So here we go, we've got this. What's missing is there's actually like a ring of proteins, like dynein, sometimes involved. It's a motor protein with actin filaments. But you get a little ring that forms around right where everything is bent in, because the clathrin, when it's built, it makes everything bend and close off, and then a little ring pinches that little last bit off, and then eventually you have this vesicle. And here we have, on the vesicle, we have these little motor proteins walking along a cytoskeleton and moving it to the next place. So we haven't had that in any of our pictures, the cytoskeleton helping move those vesicles yet. We've kind of just seen them just floating from the endoplasmic reticulum to the cis Golgi and stuff like that. But there is movement on the microtubules, or on the cytoskeleton. All right, so here it is. I don't know if you guys can see it back there, but this is the what this looks like. Okay, so this is at the membrane, what it looks like to call the clathrin to the membrane so we can start bending the membrane. Okay, so we have in here some of the molecular players, just to point them out. We've got ARF and an ARF-GEF. So we've got GTP in here. Remember what GTP does? It's an on switch and an off switch. Okay, ARF is basically, we've seen, it's a relative to the RAS and RAN proteins. We saw RAS when we were moving things into the nucleus. Just as a reminder, you want to go back and look at those. But it's RAS or RAN? I'm forgetting now. But they're movers. They move things and they call things basically. So, anyways, ARF, when it's off, is GDP bound, and when it's on, it's GTP bound. So, following the diagram, you have to have a guanine exchange factor that turns it on. So pulls the GDP off, puts the GTP on. Once this is on, it's able to bind an adapter protein. These are cool because this will actually bind. So the adapter protein binds a receptor that's going to bind whatever that cargo is that goes into that vesicle. Okay, so it's all of these things come together. Let me do it backwards for you. So your cells, you you end up with a protein and it needs to get into a vesicle. That protein is going to bind a protein-specific receptor on the surface of the membrane that's going to become the vesicle. Okay, that has to be bound by some adapter protein. So it's a transmembrane protein that binds a peripheral protein. It's an adapter. So AP1 is one of the more famous ones for clathrin. AP1 is a clathrin adapter. Okay, but that adapter protein is only able to bind the receptor bound to the cargo with the help of a GTP bound ARF protein. Okay, all those layers of regulation, all the pieces gotta be there to make sure we get binding in the correct membrane and eventually build the correct vesicle, right? Okay, so here we are. We've got all the things together, and now our adapter protein is it's on because it's bound to the GTP bound ARF, and now clathrin is able to bind it. So clathrin is a triskelion, is something that we call it. So it's literally like it's a really cool filamentous protein, and there are 3 pieces that radiate from like a center point, and they're really neat because once they're recruited here, then all this stuff gets out of the way. Or the AP1 stays bound, but it eventually it folds itself on the membrane. And when one is recruited, another one comes on and binds, and it like nucleates, and then it grows out like a crystal, all the way around the membrane. Okay, so here we go. It's clathrin, the coat assembly here, so you can see all of these things. 3 little things come together, and they form this like honeycomb type of structure, this cage. We call it the clathrin coated cage. So here is a clathrin coated vesicle. It's budded off, it's able to move, but at this point it's unable to fuse with any other membrane or wherever it's supposed to go because those coat proteins are in the way. All right. They're not meant to float around in vesicles, though. The proteins are meant to go somewhere, either be secreted out or stuck into the plasma membrane or into the lysosome, right? So they have to have another mechanism to get there. Okay, so let's follow this one. All right, so there is another R family protein that you're gonna need to remember. I know all of these R's in this, but the RABs. So RAB, there are several members of this family, and this is a tethering factor for membranes. And it's really interesting because there are RABs, like RABs are going to regulate the ability for all kinds of different vesicles to interact with different membranes. Okay, so there's like specific RABs in the early endosome. There's specific RABs on the late endosome. There are specific RABs in the lysosome, and they're specific to that membrane. Okay, so how does RAB work? So RAB actually will be in the membrane of this vesicle that we pinched off. There are a couple of other proteins that are in the membrane that has been pinched off. Notice that the coat is there. That's what the red dots are. So that's the clathrin. Okay, so these are the RABs there, and the snares are there. These are really cool. These are another long filamentous protein, but they're all kind of folded up while this thing is being walked along. Okay, but the snares do kind of what their name says, and they snare they intertwine with snares of the membrane that they're getting ready to dock with. Okay. So here is our target membrane. Here's our vesicle that just walked along. We have a RAB there. The RAB binds something called the tethering factor on our target membrane. It's basically a receptor that's receiving the vesicle. Okay, and the ligand for the receptor is the RAB protein. All right, so the RAB is surrounded by these snares, and once this binds, then these things are able to open up and then wrap around each other. So here we have that wrapping around shown here. In the process of like ratcheting or twisting around each other, the RAB protein scoots out of the way. So it gets laterally diffused in the membrane out of the way. The next thing that happens that's not shown here is the coat has to disassemble. So the clathrin proteins do need to fall off because we cannot fuse a membrane with a protein-coated vesicle. So those go away, and then these continue to wrap into each other, and they pull the membranes to each other, and then the lipids just start to flow into each other. So they fuse, and then these things just continue to basically rip that vesicle open and push it in. Does that make sense? So can you imagine these things moving to either side of that image and pulling the membrane, popping it open and infusing it into that surface of the new target membrane. So that's fusion at the destination. Okay, so a couple of new proteins that we learned about: ARF, the ARF guanine exchange factor, the adaptor protein, and the clathrin to build the cage. And then we move it to where it goes. When we get there, we've got snares and RABs that help us to tether and fuse the membranes together so that we can deliver the cargo. All right, our lysosome, just a reminder, I kind of started off with this. So they are that membrane enclosed organelle that has all those dangerous enzymes in it. They're, basically the stomach of the cell. They digest all the stuff, and they have specific transporters in them to make sure that the acidity is maintained by transporting in H+ protons. So pH 5 is their average pH. We never want that thing open, right? So now that you've seen how we can fuse the membranes together without anything spilling out, right? That's an important thing. But remember that the lysosomes digest things that are taken up from the surface. So there's going to be two directions that things are coming to converge on the lysosome. So you've got endocytosis coming from the surface of the cell, and then you've got the endosome, and then you've got the enzymes, all these little popcorn-shaped things, all of these things coming from the Golgi, fusing with the endosome, and then eventually the lysosome. Okay? So common destinations, there are some common destinations for things. This is a really grainy image, so I have to apologize for that. But here is showing us, here is phagocytosis, or I keep saying endocytosis, but really when the cell is eating something, it's phagocytosis, right? So here's a picture of that. We're taking something from outside of the cell, inside of the cell. Here's our lysosome, and we put them together. We have phagolysosome, okay? But then never trust a PowerPoint, especially now you're a molecular biologist, because all the other stuff is missing now, right? So, but you have a picture of where the enzymes come from, that too. So this is also a reminder that it's not just things that are coming from the surface of the cell. We have things that are coming from within the cell. When our mitochondria is done, this is how we can destroy the mitochondria. A membrane can be built around these things and brought to the lysosome. So similar mechanism for these things, okay? Fusing them with a membrane, wrapping them around a membrane, clathrin, COP, those things, okay? This is one that is showing you outside of the cell. So we have, here's some stuff that's coming in. This is the endocytic vesicles. Here's that recycling endosome. So just a reminder that there is an inward movement and it's vesicular transport as well. I might have a better picture here. So this is this was drawn by my late mentor, and it's just a really kind of a cool thing to just show you the movement from the surface of the cell into the cell. So here is our cargo. We pinch it off with clathrin. Clathrin has to be uncoated. We have an early endosome that has a certain RAB protein that will be embedded in the membrane. We have eventually the late endosome and the lysosome. This is actually an experiment that I did. These are my pictures. So this is showing you where the RABs are going to be, and remember that each membrane is going to have different RABs to help facilitate the proper movement in the right direction. So typically RAB 5, you'll find it sticking in the membrane of our endocytic vesicle. RAB 5 follows all the way to the early endosome, the late endosome, eventually, because of just the way the membranes are matured and things are targeted to it. The late endosome has RAB 7 in it, so there's lots of different RABs. In this experiment, what were we doing? These are pancreatic cancer cells, and we were looking at whether or not We could use these mutants of Rabs. We had the wild type DA and DN. This means that we changed a nucleotide so that they would not leave. They wouldn't leave and like go to the next vesicle. They wouldn't mature into the next vesicle. So here what you're seeing is we've got proper movement. We've got these little vesicles because they're moving along, right? And these ones, this is the one that prevents the movement afterwards. So the vesicle that is holding the Rab5, this thing, gets bigger and bigger and bigger. We never move on to the late endosome. So you see these big circles. And this is an early endosomal marker. So we were able to prove they all stuck together, right? So the green went with the red. So our mutant was doing what we wanted it to do. And this one is one that actually prevents the this direction. So we had this like really diffuse, the Rabs were not localized to the membranes like they were supposed to, so it's like just diffused through the cell, probably needs to be degraded. And then the early endosomes were all messed up because they weren't linking up with the Rabs at all. So that's a fluorescence microscopy way where you can watch trafficking. You can mutate those snares. We've done that too. Mutate the snares, mutate the specific Rabs, and you can, this isn't a therapy. We ended up using this to figure out where a different protein was going. Like, could we stop the proteins in the early endosome and see some different outcome? Okay. All right. So some test questions we can play with, just to get you back to thinking about some of the things we've talked about. It's been a little while. I don't know if I brought this one in yet, but ubiquitin. So we've been talking about protein modification and stuff like that. So randomly grab this one. So ubiquitin is a small protein that is added to other proteins in multiple copies. What generally happens to a protein that gets ubiquitinated. These long words. Does it move to the nucleus? Is it delivered to the proteasome? Does it enter the lumen of the endoplasmic reticulum? I heard it. It's B. It's the proteome targeting the targeting signal for the proteasome, right? So we've learned a lot of targeting signals. We've learned like a nuclear localization signal. We've learned the signal recognition particle and stuff like that. So you might want to, that might be a flashcard thing you want to do, or just maybe write down what are all the localization signals that we talked about. So we spent a little bit of time talking about membranes, right? So which of the following best describes the membrane bilayer in an animal cell? We talked about how the membrane was built and how there were flipases, right? So, okay, is one leaf, and we did not talk enough about cholesterol, but one leaflet is cholesterol and the other leaflet is only phospholipid. Is that true? No. There's cholesterol. It can't be that way because the cholesterol is really rigid, right? So it's kind of diffused through the phospholipids. Each leaflet has cholesterol and a random assortment of phospholipids. Remember we talked about how the phospholipids are specific? We had ethanolamine, we had right? So are they randomly assorted? And then C is each leaflet has cholesterol and a characteristic set of phospholipids, or each leaflet has a random assortment of phospholipids and neither has cholesterol. C, yeah. So the hard part is that one thing I thought maybe, I brought this in here because I'm like, we didn't talk enough about cholesterol. The hard part might have been like, what this cholesterol thing is, where is it? It is on either side of the lipid bilayer membrane. But remember that you're going to have phosphatidylserine always on the, I didn't ask you to remember the specific ones, but just to give you an example, phosphatidylserine is always pointing cytosolically. And then you'll have other phospholipids that'll be pointing to the extracellular side. So there is a difference in the phospholipids that point out and in, right? At the smooth endoplasmic reticulum, that's what we spent some time talking about in this lecture, right? In the smooth endoplasmic reticulum, we have the building of these lipids, right, with the glycerol, the 3-carbon glycerol, the fatty acid chains, on one side, because all the machinery is on one side, and then they have to be flipped so that we have, you know, equally dense 2 sides of the membrane. Okay, here's one. After the SRP binds the signal sequence, what will happen to the ribosome translating this protein? Is it gonna terminate the translation and release the protein? Will it become a membrane-bound ribosome? Or will it move through a nuclear pore? Or will it be secreted from the cell? So we're specifically talking about a protein here, right? That's in the question. So after the signal recognition particle binds its sequence on what side? The N-terminal side of the polypeptide as it's coming out of the ribosome. What happens next? It's stuck to the ribosome, right? The SRP binds the N-terminus of that little chain as it's coming out of the ribosome. And this would be, either way, for co-translational transport, the ribosome then moves to the membrane, and it's stuck to the membrane of the endoplasmic reticulum, right? Because there's the SRP, and then it's bound to the N-terminus of that polypeptide chain, and then sitting in the membrane of the ribosome, right next to the translocon, is the SRP receptor. Okay, so another picture to draw out, moving this stuff in. Don't be defeated. I see like everybody's like, oh. Don't forget there is that study guide posted. So stuff to, I think that will be really helpful. Okay, we've got about 10 minutes. I know it's a Monday. And listening to me talk isn't like everybody's favorite thing. Well, we could either start this, but I feel like we've got lots of time to do this. Do you guys want to have a few minutes to talk to each other about your bioinformatics stuff? Okay. Let's go. We'll pick this up on Wednesday. You guys talk to each other about where you're going with your bioinformatics.